tools that facilitate and accelerate genome evolution. In general, when speaking of evolution, we need to distinguish at least three categories of structure and function for molecular interactions. 1. Molecular interactions may contribute directly to the survival and reproduction of the individual. 2. Molecular interactions may serve as tools that generate structures and functions during the development of the individual. 3. Molecular interactions may serve as tools that contribute to genome evolution. The genome represents a platform containing several innovations that serve as tools for 1. Storing and transmitting genetic information from cell to cell and from generation to generation. 2. Accelerating and facilitating further genomic evolution in a variety of ways. 3. Linking evolved, heritable information from the genome to developmental processes that generate complex biological structures and functions. Every day, laboratory biologists throughout the world expand the depth and breadth of our knowledge concerning the interactions and functions of specific sequences in the genome. Most of the new discoveries are focused on the complex chromosomes of eukaryotic cells. How can we index, leverage, and keep track of these discoveries? The ENCODE consortium represents a success story for international scientific cooperation. Here's a description from their website. The ENCODE, Encyclopedia of DNA Elements Consortium, is an international collaboration of research groups funded by the National Human Genome Research Institute, NHGRI. The goal of ENCODE is to build a comprehensive parts list of functional elements in the human genome, including elements that act at the protein and RNA levels, and regulatory elements that control cells and circumstances in which a gene is active. ENCODE clearly demonstrates that genomic DNA sequences perform a broad range of functions. Both non-repetitive, complex sequences and simpler sequence elements with varying degrees of complexity contribute to these functions. The 1970 concept that much of the genome consists of junk DNA should be replaced with a more modern view of a scrapyard.